audio? Okay. All right, can you hear me? Hello? Yes?
Yes.
You sound great, John. Welcome.
Thank you. Sorry, a little crazy down here at the station trying to get this up and running, but I think we're good. Test, 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 test. Everybody hear me okay?
Yeah, you sound good, John. And we've got our audience joining us here. We've got hundreds of people tuning. While we're getting set up here, if you want to go ahead and open that chat and let us know where you're watching from, we would love to see where everyone's tuning in from. And John, when you're ready, go ahead and take it over.
Okay. Thank you very much. I appreciate it. I want to just double check one thing before we start. We have all our guests, correct? Looks like everybody's on the line with us. I think so. Okay, I'm going to get started. Well, good evening, and thanks so much for joining us. Welcome to Cut, Paste, Delete, the Ethics of Gene Editing and Humanity's Hereditary Future. I'm John Daly. I'm the health reporter with Colorado Public Radio, and I'll be moderating tonight's discussion. Thanks so much to all of you for joining us. I'll introduce our distinguished guests in just a moment. This is a free public webinar. It's presented by the Center for Bioethics and Humanities at the CU Anschutz Medical Campus, the Denver Museum of Nature and Science, and the Institute for Science and Policy. This event is also being live streamed via Facebook and YouTube. Audience members were invited to submit questions for the panelists upon registering. We'll get to some of those later. Unfortunately, we won't be able to get to all of those questions, but we look forward to trying to answer some of them. Tonight, we'll be talking about CRISPR, now one of the world's great acronyms, of course. That's the revolutionary gene editing technology that's taken science by storm. It uses microbial immune response to selectively snip DNA in living organisms. The technology's broad applications are breathtaking for modifying and enhancing food or fuel, therapeutics, cancer treatments, and more. Less than a decade ago, researchers published their initial landmark paper in the journal Science. Then last year, those same CRISPR pioneers, Jennifer Doudna and Emmanuelle Charpentier, were awarded the Nobel Prize in Chemistry. You know, it's no understatement to say that CRISPR has quickly unlocked a new world of biomedical possibilities. A recent New York Times story summed it up with this headline: once science fiction, gene editing is now a looming reality. But with gene editing's newfound ease and speed also raises all kinds of ethical uncertainties. And that's what we're going to be talking about tonight. Tonight we have a panel of expert guests, including Hank Greeley. He's the director for the Center for Law and Biosciences at Stanford Law School. We have Rosario Isasi. She's the research associate professor at the Miller School of Medicine. That's affiliated with the University of Miami. And Michael Zaretsky, he's the director of the Colorado Fetal Care Center and Maternal Fetal Care Unit. That's at Children's Hospital Colorado here in Colorado. Hank Greeley, let's go first to you. I want to ask I want you to start explaining what this acronym CRISPR means. You have a new book out recently that gets into this. CRISPR People, the Science and Ethics of Editing Humans. Tell us what CRISPR does and explain why it's considered such a breakthrough research.
Happy to. CRISPR is an acronym. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. So I think everybody can understand why we're all happy that somebody came up with a nice, easy to pronounce acronym for CRISPR. I actually, in the course of preparing my book, emailed to the two guys who came up with the acronym, a Spanish microbiologist named Francisco Mojica and a Dutch microbiologist named Ruud Janssen. And they were both doing this sort of similar stuff with this weird bacterial thing that they didn't understand. And they were calling it different things. And they got together and said, look, we should pick a name. And so they actually picked a variety of acronyms. They went through the list to see what acronyms were currently being used in the biomedical world. They looked at something called PubMed, which the National Library of Medicine runs, to see what acronyms it recognized. That threw out some of their choices. They ended up with CRISPR, which I thank them for, because it not only is easy to pronounce, but it verbs and it adjectives so easily. You can CRISPR something, something's CRISPR-ish, something's crispy. Just a great word for it. I do want to modify a little bit something John said in starting. Jennifer Doudna and Emmanuelle Charpentier did wonderful work, but they were building on the work of scores, if not hundreds, of earlier researchers, including in particular, I think, Mojica, but also like people at the Dannon Yogurt Company played an important role in understanding how to do this. But most fundamentally, CRISPR was not invented by humans. CRISPR was invented by bacteria about 3 billion years ago. Now, they don't know who the bacterium is, and the Nobel Prize Committee requires that the person who gets the prize is a living human, so they couldn't give the Nobel to the bacterium, but some bacteria came up with a way to defend itself. CRISPR is a variant on kind of a bacterial immune system. It turns out that viruses don't just infect us, they infect bacteria. We tend to call those viruses that infect bacteria bacteriophages or phages, but they're viruses. And CRISPR was a way bacteria came up with to break up the DNA in those viruses. Now, we talk about this as cut and paste and something else. I forget the exact title of tonight's program, but I think it really should be find, hold, cut, and replace, because that's what CRISPR in its fully worked out form does. A key piece of CRISPR is a piece of RNA, which is the very useful and crucial cousin to DNA, that matches a stretch of DNA, complements a stretch of DNA. So if you've got a stretch of DNA that says AAATTT, and then you put out a stretch of RNA that says, UUUAAA, that stretch of RNA is going to bind to, it's going to find and hold on to the complementary stretch of DNA, because A's only connect, in DNA, A's only connect to T's. In RNA, they're used, so I should have used the other pair. G's only connect to C's, and they stick together like a lock and a key. So there's a piece of RNA, it finds a piece of DNA you're interested in. Then there's an associated protein, the most famous of which is called Cas9. Cas, not so creatively, stands for CRISPR-associated protein, number 9 in this case. It makes a cut through both strands of the DNA at the spot where the RNA is holding onto it. So right now, it's kind of like Microsoft Word's find and delete function. So if there's a manuscript that spells my name Greeley, which is not the right way to spell it, I can say find Greeley and take it out. And that's what basic CRISPR does. And that's what the bacteria used it for, to kill chunks of DNA in the viruses that were invading them. The next trick is to add to this whole complex some more DNA that, when you get lucky, and which is a high enough percentage of the time, will slip into the hole made when you've cut out the piece you wanted to cut out. So that's like replace in Microsoft Word. Find all the Greeley's, cut them all out, and replace them with Greely. And when it works right, that's the basic use that we're most excited about with CRISPR. I have to say, though, it's only 9 years, it's less than 9 years since they published. CRISPR is turning out to be kind of a Swiss Army knife with tens or scores of different possible uses. This is the most famous one, but it's by no means the only one. So what could we use CRISPR for? There are 3 important ways we can use CRISPR. We do use CRISPR. One is just straight for research. You want to change genes to see what happens. You want to change the genes in human liver cells that you've got in a dish to see that if you knock out this gene, does it affect how those cells deal with alcohol, for example. Straight research uses CRISPR is wonderful for making changes you want in genes, particularly knocking out or making genes non-functional. Second, you can use it to replace, to cut out or replace genes in living people. So if I had sickle cell anemia, it's a disease, a blood disease caused by a genetic problem, genetic mistake that's in all of my cells, but it's only important in the cells in my bone marrow that make blood cells. There are people working on being able to take your bone marrow out, use CRISPR to replace the ineffective form of the globin gene with the right form of the globin gene, grow those cells up, put them back, and cure you of sickle cell anemia. And, I think we're probably close for that. And we'll hear later for lots of diseases. But that has the function, that has the characteristic, it affects me, but it doesn't affect my kids. It's in my bone marrow cells. It's not in my sperm. If it's not in my sperm, it lives and dies with me. If it's not in Rosario's eggs, it lives and dies with her. If it's just in the body cells, the so-called soma or somatic cells, that's an end. The third use is germline. The germline is the eggs and the sperm. And what makes the germline special and particularly controversial is that's when it passes down to the next, can pass down to the next generation and potentially the next generation, the next generation, and the next generation, so on ad infinitum. That's the one that's been getting the most attention. And I think I will leave it at that for now, having taken probably longer than you wanted.
All right. Thank you very much for that introduction, Hank. I really appreciate it. Let's talk just a little bit before we go to our other guests about a man named He Jiankui. In fact, the first chapter of your book, I have it here, CRISPR People, on sale at a bookstore near you or Amazon.
Could confirm, I didn't pay you to say that.
No, you
thank
did.
You.
You did. Yeah, you bet. So in fact, the first chapter of your book is titled "Just What Did He Jiankui Do?" So maybe can you answer that just real quick and tell us about why this has been such a global controversy, and then we'll get it right.
Sure. So remember those three uses: research use in cells, use in living people to change their genes, use that changes eggs and sperm. He Jiankui, and anybody who speaks Mandarin, please forgive the way I butchered his name. I've never been able to get anywhere that doesn't elicit laughs from native Mandarin speakers with pronouncing that name. JK is what he went by in the US for reasons that I think I understand. What he did was CRISPR embryos, human embryos, which he then transferred into women's wombs, the women who had provided the eggs and whose husbands had provided the sperm, in the hopes that some of them would become babies. As it turned out, 3 of them had become babies. 2 at the time all of this broke in November of 2018. This caused a huge uproar for lots of reasons. This is maybe the most reckless, stupid, annoyingly bad research I've seen in decades and decades of following this stuff, because the risks to these babies were enormous and unknown. He hadn't done this with monkeys or other things, and he hadn't checked to see whether it affects the embryos. And in fact, one of them, he knew that he hadn't made an effective change. He didn't make the changes he wanted in either of the first 2 babies, but he moved the embryo, and he knew that before he moved the embryos into their mother. And he did it anyway. So this was risky, unethical, bad research. It also, though, was the first time anybody's tried and succeeded, we think, in changing the cells of an embryo. And the embryo here is important. These are early embryos, like 6 days old. You change it at that point, you're likely to change all the cells in the body. So if you change bone marrow cells, you're just changing bone marrow cells. But if you change the cells in an early embryo, you're probably changing the cells that will later become brains and livers and eggs and sperm. So these two little girls, the third baby we know almost nothing about. It was born 6 months, 8 months later, and we know one sentence from a government press release, a Chinese government press release, saying it was born. These two little girls could pass on these changed, never seen before in human genes to their descendants and their descendants and their descendants. And that has many people upset. Some people think that's a line we should never cross. Some people think, well, maybe someday for good reason, if we know it's safe and effective, we could try to cross it for some good reasons. But very, very few people, Hu Jiankui, who's now serving a 3-year prison term, we think, so it has been announced, is maybe one of the few people in the world who thinks this is a good idea to do now. There's an overwhelming scientific consensus that it's not shown to be safe, it's not shown to be effective, it's criminally reckless to do it now. And some people would say you should never do it, and some people should say if it's proven safe enough, there may be some good uses for it. And that's where the debate stands right now.
All right, that's a great introduction, and I think we should segue now to Michael. Michael, what do you think are the implications of what we've just talked about, both on the perhaps dangerous side and then also on the promising side? And this, I think you could speak about some of the research that you and your colleagues are doing.
Sure, yeah, thank you for having me on the panel this evening, but I think it's important to start off by saying that my specialty is in maternal fetal medicine, and my expertise is in fetal diagnosis and therapy. I'm not a molecular biologist, but I wish I was actually, but I work with talented researchers on our campus who are experts in the field and with CRISPR in particular. So hopefully I'll do this some justice this evening. In my field, the current state of the art is that we're very adept at making a diagnosis of fetal disease by ever improving imaging capabilities. And it's not uncommon for us to sequence genes in the whole genome sequencing for fetal diagnosis. We actually have a fetal precision medicine group for this purpose. We actually have a couple of bioethicists that sit on that panel. And what we've done is we've extended what already done in the neonatal intensive care units where they use genome sequencing, and we have extended that back into the prenatal period. And the cost of this has really come down, and the speed of testing has really improved over a relatively short period of time. And when we make a diagnosis in utero, there are only a finite number of interventions that are available that we can make an impact on the prognosis for the child. And for most diseases diagnosed before birth, the only intervention we have is to plan for a multidisciplinary approach to treat neonatal and pediatric disease, which fortunately for many is enough, and that is what we do. For most genetic diseases diagnosed in utero, such as cystic fibrosis or many metabolic diseases, you simply have to wait until delivery to begin treatment. And for many of these genetic diseases, the manifestations can be quite severe and even lethal prior to delivery. And so now comes along CRISPR, and there's this immense potential to prevent or even provide a permanent cure for these genetic diseases. And because of this new technology, I had the opportunity to approach experts in gene editing from actually 3 different locations. One, our campus at CU Anschutz at the Gates Center for Regenerative Medicine. It's the Anna Bilasova, Igor Kogut, and Sean McGrath, Colorado State University, and their large animal lab with Russ Anthony, and National Jewish Hospital, Jen Masuda, all experts in this area. And I have to say, coming from this foreign topic, to me, it was a bit intimidating, quite humbling experience. But what I discovered, there's a real interest of collaboration between bench and bedside, me being the bedside. So currently our group is working on cystic fibrosis in the lab and subsequently in a large animal model, targeting the most common mutation to see whether we can efficiently edit this in utero. And we're making slow but steady progress, but there are multiple hurdles and challenges. And I emphasize that we have a long way to go until we get to the point where hopefully we can use this and apply this technology for genetic disease diagnosed before delivery. So our criteria for this, for in utero treatment, would be that we have to make a reliable prenatal diagnosis. And if we do treatment before birth, it's got to result in a healthy child and prevent serious morbidity, mortality, and that the current treatment for the disease reliably results in a poor outcome for the child. And fetal intervention must improve this. Fetal intervention also has to be low risk for both the mother and the fetus. And lastly, what we are doing is targeted somatic cell editing and not germline editing. And we'll be trying to avoid complications where you have off-targeting effects. So if you were targeting that CF mutation, but it ends up making cuts in DNA far away from this area, that we want to avoid that. And one of the areas we want to avoid is germline cell editing that Dr. Greeley really did a great job at describing. And we're going to discuss more about that at length and what that means. So I guess with that in mind, I'm going to hand it back to you, John.
Okay, great. Before we move on, maybe you can just, again, sort of, it seems like a key point for the audience to understand here is this difference between germline and somatic, and kind of the layman's terms, I guess the somatic would be dealing with just one child, just one person, whereas germline would be something that would be heritable and passed on from generation to generation. Am I correct in summing that up that way?
Yeah, so like what we're trying to do is when we're targeting cystic fibrosis, which affects the lungs, the intestine, the liver, et cetera, that's what we want to target. And we want to avoid targeting cells that are from the germline, the stem cells that turn into sperm and egg, and that would be propagated from generation onward. And if you make errors in that, that can have some serious ramifications. So we really just want to be a targeted approach to affect those organs that can really make an impact for a child.
Just so we're all clear, this is all very much still in the research phase, and you haven't actually delivered a baby through using a CRISPR method. How far out is that kind of work, either here in Colorado or elsewhere in the world?
I mean, all I'm aware of is this work in the mouse model, different animal models of disease, which, with varying degrees of success. There's actually a whole organization called iFetus looking at stem cell and gene editing in utero that meets yearly, and it's pretty remarkable what some of these researchers are doing. It's hard to put a timeline on this. I'm not sure. I mean, the way things have accelerated, are we talking a decade, 15 years? I'm hoping that we'll have therapeutics available by the end of my career. I think sooner the better. I think it could be done. The technology is pretty amazing. I think the biggest hurdle is actually packaging the gene editing tools and delivering it with high efficiency to the tissue you want it to go to. And I think if you can overcome those hurdles, we'll have something in the pipeline.
All right, great. Well, thanks to both of you. Thanks, Michael. I think you've gotten us up to a really great start here. Rosario, let's turn to you now. You're a lawyer and a bioethicist. Let's kind of broaden the discussion out. What do you see as the primary policy implications of all this science?
Well, first, I will say when we talk about policy, we are talking about a big framework. Imagine you're assembling a puzzle and you have different pieces, right? Some parts will be laws, as we all understand, that Congress, Parliament pass a law, but also part of policy is professional guidelines, best practices, how societies like clinicians societies and research society, the American Society of Human Genetics and others regulate practice, but also we include principles and values that society holds. So what are the implications for gene editing from the policy point of view? They're humongous. One of them is the distinguishing between which kind of application are you dealing with, and there's a lot of variety across countries. Some of them will prohibit just using gene editing or CRISPR, or regardless of technology, in the germline, regardless of the purpose, is what we call it top-down, right? But others will say, well, you can use this technology, but depends, right? And it means that you can conduct research in vitro. You can conduct, in some countries, even preclinical research But the hardest stop so far, and then we can talk later about the ethics and the implication of this policy, is that some professional organizations have said, well, maybe one day we cross that threshold and we will allow the implantation of an embryo that have been gene edited. So if I'm gonna tell you, I told you that it looks like a puzzle, right, that we're gonna assemble, but if I was gonna classify the different pieces of the puzzle according to how restrictive or prohibiting they are, so how tight they are in regulating the technology regarding application, it will look like a Christmas tree, right? You will see all these colors and all these things. So there's no one single policy. Something that is very interesting, I would like to say, and connecting with what Hank and Michael have said about history, right? Whether CRISPR-Cas9 gene editing sits on the shoulders of a generation of scientists who laid the background, policy in the area does the same. In terms of gene editing in the germline, on the embryo and the human being, the policies exist for a very long time, and they were at their peak in the 1990s and the 2000s, when we were discussing issues such as cloning, genetic engineering, etc. That's what, even now, most of the laws are. They're outdated and they come from that. For somatic cells, so what do we do in the tissue to human beings that they're not having intergenerational effect, more of the policies and the laws are the regulars that govern gene therapy and other type of human research or clinical trials. So they're more standard, if you wish, right? To put it in lay terms. However, since JK experimentation, because I'm sorry, I cannot call it a clinical intervention, since that case of human experimentation, they have been even a more uproar, if you wish, a systemic effect that everybody with a voice, everybody with an opinion will have an opinion translated in a policy. That means that a lot of professional organizations and other bodies have started as a pronouncement trying to regulate what kind of application we will have using gene editing, but always making this distinction of regarding where are you doing the intervention, right? In the somatic level or the germline level.
It sounds like the laws and the policies are nowhere near where the science is in terms of how quickly this is all evolving, right? And in the United States, would you say we don't have much law anyway from Congress governing this?
Well, the answer is quite complex, because as this is a puzzle across the world, even the most, I would say the better model of regulation, the more tight that is the UK, there's always what we call the lawyers' loopholes, right? Little gaps where the spirit of the law, what the legislator intended to protect or govern, right, or regulate that behavior, or the letter of the law doesn't apply clear cut. So I have done several studies that we look and they said, oh, the law in principle is very tight. And then we said, well, no, there's an exception here. So the laws, even the most perfect, they're not right tight. I don't think we necessarily need to change the laws. Most of them were reactionary at that time, and we need to understand better whether a fit for purpose and understand that there's a whole cosmos, right? That one single law will not be sufficient if we don't look at the different stages of research, if we're not written stages of clinical application for gene editing. The US, we can have a long debate about how imperfect the system is, how patchwork it is, because not a single law. Most of the provisions come in restrictions for funding. But overall, I would say there is a general consensus across countries that conducting research and even clinical applications in the germline, you should work with care, approach with care. If it's not a big no-no, it's a maybe, but with a lot of caveats.
Real quick, did the case of He Jiankui in China speed up the desire of policymakers and lawmakers and researchers as well, scientists working in this realm? Did it speed up the desire to start to make some policy that could give some guardrails to this kind of work?
I think, if anything, that started since CRISPR became a trendy issue, has become a breakthrough. Yes, the Chinese case brought it more into attention, but not only the laws, because what he did was illegal in China. And I was even at that time consultancy with the Chinese Academy of Science. I was working with China. It was illegal. The issue is, and continues to be, what we call governance. So how you can have control and oversight what researchers can do. The law cannot do much if people want to disobey it. So the issue is how we enforce it, right? And another issue that we also will discuss is how a national legislation is not sufficient, because genetics as science is a global enterprise. And if anything, JK case shows how there's always international actors. And if we don't have this level of international policy, there's not much what we can do.
Okay, that's great. Yeah, very interesting all the way around. Let's take this kind of as a jumping off point to discuss another book that you might have read, or some of you have seen at the bookstore. This is The Code Breaker. This is about Jennifer Doudna and her work by the great Walter Isaacson, who's written biographies of Leonardo da Vinci and Steve Jobs. And he notes this newfound ability to make edits to genes, that it raises all kinds of fascinating questions like, should we edit our species to make us less susceptible to deadly viruses, like one that's going around the world today and has been for the last year? Or should we eliminate dreaded disorders, or things like maybe being short or being depressed? Hank, really, that's kind of a slippery slope, right?
Yeah, although to say something to slippery slope doesn't necessarily tell you what you should do with it. I think we live on slippery slopes all the time. I have been known to go faster than the speed limit. That's breaking the law, but it doesn't necessarily mean I'm going to become a mass murderer, but it's a step on a slippery slope. I think that the issue of what we should use genetic technologies for is one that has some broad agreement in it, but some real disagreement in some places. Very, very few people disagree with the idea of using genetic technologies to fix things that are clearly bad diseases. So one of the worst of many nasty, happily rare genetic diseases is something called Tay-Sachs disease, where babies are born healthy and their brain cells die, and they die after a few years. It's a terrible disease. If we've got a genetic intervention, either like if Michael's work pans out, either fetally or post-birth, that can avoid that, nobody's going to be complaining about that. Then you get to things like, oh, I have slightly different color vision than most people. It's a genetic trait. About 5 or 6% of American males see greens a little less intensely than apparently the rest of you do. I have no idea what you guys see. Should we use it to fix that? I am fortunate enough to have genes that give me, great white hair at an early age. Should we fix the rest of you so you get those kinds of genes? You move into cosmetics. At the far end of this, we get questions about making people smarter or better athletes or better musicians or better mathematicians, or give them a better personality. I frankly am less worried about those because we don't know how to do it. We know hundreds of genes that are powerfully associated with intelligence, in the sense that if the gene is broken, the person has very low intelligence. But we know almost nothing about genetic variations that lead to above average intelligence. We know things like good childhood nutrition and avoiding childhood disease and having your parents read to you. Those help intelligence. And I think one of the areas I work in is neuroscience and law. I think neuroscience makes genetics look like child's play. And it's going to be a long time, if ever, before we fully understand, yes, there will be genetic components in some of these behavioral issues, brain-based issues, but wow, they're going to be complicated, involving hundreds of genes and lots of environmental stuff. So yes, if you want to use it to make X-Men and Supermen and a genetically dominant inherited caste, very few people are in favor of that. probably a few teenage boys locked away in cellars someplace, but very few people are in favor of that. If you want to try to cure people of Tay-Sachs disease, babies of Tay-Sachs disease, lots of people are in favor of that. I think the diseases, particularly the ones caused by a problem in one gene, that's where we're most likely to see the activity. And there will be some things like, hair color, which is not one gene for the most part, but sort of light versus dark. You can do something there. There will be some pressure there, both with editing, but also with selection, because we can tell from an embryo through something called pre-implantation genetic diagnosis. We can test to see whether the embryo will have a disease or not, but you could also test to see whether the embryo is likely to be a baby with light colored eyes or dark colored eyes. Exactly what kind of light or dark we're not very good at, but light or dark we're pretty good at. And so you've got selection on the one hand, picking the embryos that have the traits you like. You've got editing on the other hand. When and how those are different gets complicated.
Yeah, Michael, do you see it that way too? And then is human germline editing inherently bad, or could it sometimes be bad? What's your take on that?
I just have a hard time getting to germline editing. If I think about patients that we see, if we know that one parent has a dominant trait, and we know there's 50% chance that the child is going to inherit that disease, we have technology currently. They can go through in vitro fertilization. They can create multiple embryos, and they can select the embryos without that specific gene. We'll just take cystic fibrosis, for an example. Both parents are carriers for cystic fibrosis, and they go use this technology, then 75% of the embryos are going to be healthy children. Some will be carriers, but they will not have cystic fibrosis. If you're looking in the future, if someone approaches this and they want to have germline editing, they have their embryos edited, undoubtedly they're going to have carrier screenings for hundreds of different genetic diseases, and you're going to identify whether those parents are carriers. And so through the same technology of pre-implantation genetic diagnosis, you're going to be able to avoid the vast majority of disease in the offspring if you do that. Now, the one problem with this thing is equity. I think I'm not the best to talk about this. I think Dr. Greeley and Dr. Azari could talk about this a little bit more, but if you look at the number of IVF babies per year, it's only 70,000 babies in the United States are born through in vitro fertilization, or less than 2% of all babies born. And so is that going to be the only population that has the opportunity to have any kind of, access to this technology? Right now, IVF is very expensive, and you add the pre-implantation genetic diagnosis on top of it, it adds another expense. Now just think about germline editing and adding that capability on top of it. So I think, in my mind, I think this would be only for the wealthy, and that's going to bring up a major issue. Right.
Yeah, that would bring up a lot of issues. Can you walk us through what it would be like to use CRISPR in a cystic fibrosis, or you can use another disease if you'd like, but just kind of briefly the steps involved from point A to point Z or whatever. How, I don't know if we need to go all the way to Z, but how would it work, in practice?
You're talking if you're doing an embryo?
Yeah.
Okay, not an expert. Again, I would have my molecular biologist colleagues on the line right now if I had the opportunity. But you would have to design the CRISPR constructs to specifically target what you're trying to edit out. So that would have to be done. And then you would have to apply those or deliver, have those CRISPR constructs inserted into the embryo, and whether that's done very early, just right after the sperm and is injected into the egg, you want to make sure that all the cells that are developing get that edit. And what we know is this is not perfect at this point in time. So what they're seeing from these early experimentations, that you get mosaic cells or chimeras where some cells get the edits and others do not. I think that's what happened with the JK babies. They were trying to edit this CCR5 gene to make these kids immune to HIV infection, and it didn't really work out in a very efficient and accurate fashion. So there's still a lot of work that needs to be done before you even get the germline editing to make it accurate and safe to do. I don't think they're currently at that point now.
Just to make something clear that you may have slipped through when Michael was talking, you've got to use IVF in order to use germline editing, because you've got to have that embryo in a dish where you can inject it with the CRISPR construct. You get pregnant, you fertilize an egg the old-fashioned way. The embryo is halfway down a fallopian tube. Good luck finding it and injecting a CRISPR construct into it. So you need IVF. Then you need the germline editing. Then you'll probably need the pre-implantation genetic diagnosis to see whether the editing worked. And this gets to, and I love the fact that it's the scientific researcher who first brought up the question of equity here, and not the two lawyer ethicists, but we get to Michael's issue about equity and cost.
You'll leave that to the experts. Yeah.
No, you called it first. Good work.
And just so that I'm clear and the audience is clear, so this would have to be at this point when IVF, and then also very early on, right? I think that, did I hear you say, Hank, like 6 days in or something like that?
Or earlier. Yeah, you actually, as Michael pointed out, there's some advantage to doing it the first or second day when there are fewer cells. So you really would like to make sure that the CRISPR construct gets into every cell by 6 days. You've got 100 or more of the so-called inner cell mass cells that will later become the baby, the medical researcher, the radio host, et cetera.
Rosario, I have a question for you. This is actually, I'm going to start fielding some questions from our audience. This comes from Corey. Corey asks, are there concerns, or sorry, this is Jean. Jean asks, does the hospital's ethics committee weigh in on each of these cases? Is that something that you think could, should, or would happen?
Well, yes. Around the world, all countries have, in different ways, but they all complain that there's always a level of institutional oversight body. Here in the US, we call it the Institutional Review Board. It's an ethics review board, or it could be in the hospital, could be in the research lab, that will vet. Yes, this will be based on the ethics and the science of the project, but most importantly, it is framed within what the country's law allows for. So yes, a hospital will have a way in, but it will be circumvented by what the national law established, what professional guidelines. But another thing is that in some instances, local institutions are played or often will have some conflict of interest. So the objectivity of judging your own peers in high-stakes science might not be the same, right? I'm thinking about the case of JK, for example, that was one of the issues, and it's a very complex case. You should read Hank's book and others that details, and we have written also book chapters on this. That there was it's more complicated, the answer, right? But yes, they might weigh in and they might stop when it's a research practice. Right? Yeah, what happens? Go ahead, Hank.
No, go ahead. You sounds like you were going where I was. What happens after?
The issue is what it will happen in the confines of the doctor-patient relationship, right? If and when this could be applied in streamlined, right? And there's authorization to do clinical applications, or in a scrupulous clinician or researcher trying to do it, we might not find out until, like JK, a baby is born, right? And the last thing I'm gonna say is that I can speak also when we talk about the ethics and equitiness, and I would like to throw it back to Hank on this, is the individual perspectives. I can speak as a patient. I lost a son on a rare genetic disorder, a baby. And the lawyer and the ethicist me says, of course, I will never do it. The matter with me, I think is, will I be presented the case as a human being with raw emotions? Will I do it, right? And this is the should question that should prevail the conversations before that what I can do is what the science allows us to do.
I'm sorry about that, Rosario, and I think that you know, this is the kind of dilemma that a lot of families would face, these very kind of questions. Let me follow up with you, Rosario, with a question from Carol, another one of our audience members. She had a question about oversight and notes that oversight is much less stringent in private fertility practices and even in private labs outside of universities and government research facilities. This is a serious concern, right? How would something like that be handled?
I think it will depend. Private IVF clinics, yes, they could have a lesser level oversight. But if the regulations, the regulatory bodies don't really check onto, like in other countries, how many embryos have been created, what are the practices doing, it will be only on the ethical and moral responsibilities of the clinicians. But again, as Hank said, what is really is an issue of feasibility, right? In institutions, academic institutions and others, and hospitals, they have more stringent rules, and it will be easier in a way to police them if we wish. But I think at the end is the free will of others. If you want to disobey, you will do it, right? Regardless of having the police next to you just trying to stop you. It's human behavior. Not that I condemn it, but yes.
So the oversight might be more challenging in a private facility, let's say.
Absolutely, absolutely. And it's then the responsibility of governments to monitor clinical practice. As we monitor Michael's, we as the government, as the country, right? As the regulators monitoring how a doctor practice medicine, I don't think we always put the same level of oversight or policing to IVF clinics. And this is a problem around the world. But also, I don't want to put necessarily in a bad light IVF clinics. Per personal experience, there are some of them, they are extremely scrupulous in the interventions they are doing, even under closed doors.
All right. Yeah, very interesting. Just want to give everybody a little bit of a time check. It's about 7:45 Colorado time, and we're planning to go till 8:30. We'll try to get in as many of these questions as we possibly can. I'm going to take this in a little bit different direction. We've got another interesting question, this one from Corey. Are there concerns that fixing one genetic defect will unleash others? Thoughts on that? Hank, let's go back to you.
Sure. But as is the case with almost every good question, the answer starts with it depends. So if you've got something like cystic fibrosis, where 90% of the genetic variations are one particular change, what's called delta F508, where there's a genetic change that changes one amino acid in the protein the gene makes to something else, actually takes it out, and causes all sorts of downstream problems. If you could just change that gene back to its normal type, we know that the normal type works pretty well because 99.9% of us have it, and it works well. If you're trying to change something from the normal type to a rarer type, you have to worry about what might be happening that you don't understand, because the one thing that's clear about genes is their functions are often not very clear. Genes almost never just do one thing. They do a variety of things in different places, and if you make it do something you like in one place, maybe it'll hurt you somewhere else. And let me use a specific example. I think it was Rosario or Michael, I forget which now, mentioned CCR5. So what JK tried to do was to change this gene called CCR5 so it wouldn't work. And it wouldn't let these babies' bodies make a protein called CCR5, which stands for something I can never remember. It turns out that CCR5 is a protein that's expressed on T cells in your immune system. And the main version of HIV uses CCR5 as its way to get into those T cells. So people who don't have CCR5, that main version of HIV can't infect their T cells. There are other versions that can, but they're much rarer. There are other cells that can get infected, but they're not quite as important for the progression of the disease. But we don't know very much about people who don't have working CCR5 cells, CCR5 genes. There are a few of them, like 1% maybe of Estonians or Finns or certain people in Northern Europe. And we know that some of them live long, healthy lives, but we don't know whether as many of them live to be to be old as should live. There's some very preliminary evidence that people who don't have this functioning gene deal with West Nile virus and influenza more poorly and have worse results from that. We just don't know. And so a lot of times you change one thing. If you're changing it from a known abnormal, dangerous version to a known normal version, you can be pretty confident. You can never be perfectly confident, but you can be pretty confident you're not doing any harm. You do anything else, you move something from the normal version to a different version, even though you think it's new and improved. It may be new and improved for one part of the body, but it may be catastrophic for another part of the body. So yes, you have to be careful. There's another issue around something called heterozygote advantage that I'll just drop that name and leave it out there unless somebody wants to come back to it. But there's another potential risk as well, involving other good things that we don't know about that maybe the bad gene does sometimes.
And Hank, a real quick question about JK's experiment and the babies. Do we know about how the babies are doing and how they've done since this, or is it just all under wraps?
We don't know squat. So JK was forced by really intrepid investigative reporting by a reporter for MIT Tech Review named Antonio Regalado. He was forced to reveal this. I think he was going to reveal it within a couple of days anyway, but Regalado broke the story that he was doing this. And he made a presentation at a world summit on CRISPR held in Hong Kong. He made his presentation on, I believe, the Wednesday, either the Tuesday or the Wednesday. It was live streamed. And he showed slides and he said what he had done. And that's the last we heard. He had a paper that he had submitted for publication that got denied. There are bootleg copies of what's supposed to be that paper floating around. We don't know how accurate it is. And we do know that what he'd said in some respects about the children was a lie. He said they were born healthy. Well, they spent at least 7 weeks in a neonatal intensive care unit, probably because they were twins, because twins often, these came early as twins often do. Was it in part because of the gene treatment? We don't know. And they're now, gosh, they're 2.5 years old, we think. The Chinese government has refused to say anything about them. It says that, I get this in part. It's respecting their privacy. The parents don't want them to become, freaks and heavily studied, et cetera. But I think you can probably release some scientific findings without invading the privacy of 2 babies in a country with 1.4 billion people. So no, we don't know. And the third baby, all we know is one sentence in a government press release that said the baby was born. That's it. We don't even know what that baby was edited for. It's very frustrating.
Michael, we've got another good question here from Sarah. What are your thoughts on conditions like autism and Down syndrome and reducing genetic diversity in society? That's another big kind of question. What are your thoughts on that?
Well, I think autism is extremely complex, and I'm not sure it's really amenable to the technology that we're speaking about. We don't even offer prenatal diagnosis for autism. It's a common question. Parents come in asking, can you diagnose my my baby with autism, and it's something we're not capable of. It's extremely complex. Now, Down syndrome, the original prenatal diagnosis was based on diagnosing Down syndrome. Advanced maternal age was a concept from the 1970s after amniocentesis was instituted, and that's a whole chromosome that's added to the genome. So instead of 46 chromosomes, there are 47. So again, there's not really anything that you can do prenatally. There have been investigators that have looked at ways to improve cognition or development in Down syndrome, it hasn't gotten much traction. So I think both of those are pretty hard to really discuss in the setting of CRISPR. But I agree with you. There's, and again, I'm going to go back to our panel guests because they're probably better at talking about this. But if everyone had certain traits, like let's take height, if everyone was able to edit the genome to improve height, then there would be no diversity. If everyone had improved intellect or physical ability, then yeah, I think it's a real problem with diversity in the population. I think that we lose something. Humanity loses something in that arena if that becomes widespread, which I don't think it will.
Here's another good question. This is Kate Callender. She's a student at St. Mary's Academy in Colorado. Thanks for your question, Kate. She asks, she says, Huntington's disease usually comes on pretty late in life, but you can still find it early in pregnancy. And could you do that and change the gene?
Yeah, I mean, that
Michael?
would, yes, yeah, I guess that's fine. Yeah,
Yep.
definitely. There's actually some significant ethics involved with testing for Huntington's chorea in utero, because it's such a significant disease. Because I actually had one couple whose the father's parents, one of them had Huntington's disease, and he didn't know his status, and he didn't want to know, but he wanted to know if his unborn baby, the fetus, had Huntington's chorea. So if I did testing, then the father would know for sure. If the test came out positive, the father would know for sure whether he had Huntington's chorea. So you actually have to go through a psychological evaluation before testing to make sure, God forbid, they find out the diagnosis, and you don't want them jumping off a bridge. So there's a lot of actually regulation with that testing. But yes, you can determine with a high degree of even early on as 10 weeks whether the fetus has Huntington's chorea. And would that be a target for CRISPR? In my book, yes. I mean, and again, this would be targeted therapy once the diagnosis is made. But hopefully you can, again, identify the parents before they get pregnant and offer to them, genetic counselor, talk to them about pre-implantation genetic diagnosis, and hopefully avoid that situation. In this case, the family didn't want to do that. They wanted to get pregnant and have chorionic villus sampling performed to make the diagnosis. But yeah, in the future, if you find out the fetus has a single gene disorder, then the hope is you can perhaps ameliorate disease or prevent disease, provide a permanent cure.
Wow. Rosario, let's turn to you, and I'm going to shift gears again. Here's another question about oversight, and this is from Michael. Could lawsuits be a method of oversight?
Lawsuits could be a tool for trying to have a remedy, right? To punish somebody, to inflict some punishment for a behavior that society condemns. Because at the end of the day, what the policy is, but for oversight is, I think, is a reactionary approach to a lawsuit, unless you want an injunction trying to prevent an action from occurring. And something interesting I would like to mention, because we are talking about lawsuits about the civil or the criminal system, that around the world, laws that regulate embryo research, from which I will apply the German gene editing, most of the time, or 99% of the times, include criminal sanctions. So whether it's a felony that you can go to jail, and also you can get a huge fine. So in other areas, Joe, of science, for example, pre-implantation genetics diagnosis, lawsuits were very successful to provoke changes in the laws. In countries where it was forbidden or was very tightly regulated, the lawsuits push the government to uphold what we call procreative liberty, so the freedom to have children, right? Push the governments to that. So it was the opposite. It was no sanctioning a behavior that we deem illegal or unethical was rather pushing the government to allow individuals to have a healthy baby.
And Rosario, here's another question, kind of takes us in a little bit different direction. How have religious leaders reacted to the potential of CRISPR technology? This one comes from Barbara, that CRISPR technology and applications. Would they be okay with somatic intervention, even if not germline?
Well, it will depend from which religions. There's so many practices and faiths around the world. If we think of the Christian Catholic faith, we know that we ascribe Christians or Catholics, ascribe personhood status to embryos, to potential human life. And therefore, they will be most leaning in condemning this type of interventions, but they will be more comfortable about somatic cell therapy, right? All those interventions that will not have this intergenerational effects, right? To go from one to the other, that you cannot inherit. Other religions don't see it that way because they see that they don't think that life starts at conception, or because of their religions have a more emphasis on science and health and issues like that. So I would say with a caveat, it depends, right? And it's as varied as the policy approaches that countries and professional organizations have. What I will ask is, what is the underpinning principle or societal value that this group holds? And this group could be a religious leaders, it could be others, that will inform how they feel about, how they think about the application of these technologies.
So I'd like to take this conversation in a totally different direction now, which is kind of, what if you had a world leader who, this is a question from Greg, who decided he's going to invest in desired traits, people, better able to fight disease, better looking, more intelligent. What would the proposed response be by other countries? By the US?
Let me talk. Let me talk.
By
I think it should.
Yeah, that is for Hank. That is for Hank.
Yeah, I want that one. I think it should be, go ahead, waste your money, waste your time and money. I think that's a fool's errand, certainly with today's knowledge and technology. And I think for the next several decades, if not forever, 50 years from now, 100 years from now, maybe he could do something that way. One does wonder what a super soldier would look like. You know, the old joke about a camel is a horse designed by a committee. Super soldier designed by a committee might be not such a good soldier, but it's not a bad question. But with the current state of our genetic knowledge, there's just no way that anything that is a useful expenditure of his country's resources.
Because it's so complicated, because there's so many pitfalls.
Because yes, and yes, because it's so complicated, because there's so many pitfalls, but fundamentally because we don't know the genetic makeup that leads to these sorts of traits that he wants. We don't know the genetic variations that lead to being smart or strong or brave or loving the dear leader. You know, there are some, it is clear that almost everything has some genetic connections, but they're usually really, really weak. So the people who are most bullish on the idea that you can find intelligence and genetic differences in human intelligence and genetic variations. And most people are not this, I'll say optimistic. I'm not sure whether I think it's optimistic or not, but are not as sanguine about this ability. They think they've found variations that can account for 1 IQ point. Not a big deal. And most scientists in the field don't actually believe that it's even that good. So the things we really care about in human behavior and human brains and in human reactions, they're just really complicated. They're complicated genetically with lots and lots of genes, usually having small effects. And if they have big effects, they're usually bad effects. And they're complicated because there are huge environmental influences and huge influences of chance. I think we greatly underestimate the role of chance in everything. There's a wonderful neurogeneticist from Ireland, Kevin Mitchell, has a book called Innate that blew me away when I read it, because he talks about, so your neurons connect to other neurons in your brain. And in a fetus, this is going on all the time. Fetuses are just neuron machines. They're just churning out these neurons. They're making connections. Most of what you do as a little kid is actually killing those connections. Some of those connections, pruning takes place, making a bunch of connections. But the connection is like, send this road to Colorado. It's not send this road to this particular street address in Colorado. And what county in Colorado that neuron gets to seems to be more a matter of chance than anything else. So chance, environment / experience, genes, they all play a role. Genes, in terms of the traits, the behavioral traits that we most think of as important and care about, they play a role, but it's not going to be very big usually, and it's going to be really, really, really hard to tease out. Talk to me in 50 years if you can find me and if I'm answering questions then.
And I would like to add something, John, is that Hank talked a lot about the technical capabilities, right? But I think that if such a dictator or such a ruler would like to start, or philanthropist start this project, I think he will have or she a hard time finding scientists that will be jump on board with it. I like to think that most clinicians and researchers still have some level of scientific integrity, and they will not do to pursue such a foolish path that it just infringes on human dignity.
We have a question here from Maureen. She asked, maybe Michael, you can handle this one. Could AI quicken advances in our understanding of this technology and how to safely use it?
That is a great question. I don't think I can answer. It's not a topic I talk about very much. I mean, AI is really in the news a lot these days, and I suppose it could help. I just don't know enough about it to comment about it. So, yeah, it's a great question.
So sometimes some of us in the field play bioethics bingo, and AI is always one of the squares on the bingo thing. AI, CRISPR, enhancement. And I think the fair answer, I think Michael's answer is absolutely right. He doesn't know, I don't know. I think even the AI experts don't know. It may turn out to be a huge deal. It may not.
Here's another question from Douglas. This is a little bit question of the one we had about a national leader taking this in a certain direction. Will there be rogue companies that offer something like this, kind of similar to 23andMe? Hank, do you think we might see rogue companies?
So I've had my differences with 23andMe, and they know my name and don't, I think, particularly like it, although I think they view me more as a harmless gadfly than anything else. But I wouldn't call them rogue. But I do think there are companies that are going to be pushing these envelopes. There is a company called Genetic Selection that is using a technique called polygenic risk scores, a new, somewhat controversial technique with uncertain power and accuracy to take a whole bunch of genetic variations and say, you add these 100 things together and you get 15% this way or 7% that way. And using it for pre-implantation genetic diagnosis, including for intelligence. Now they're saying that they're not going to use it for above average intelligence, but they're willing to tell you whether this particular embryo would be below average intelligence. I don't think the science supports that, but that's not well regulated. I wish the FDA would regulate that kind of thing more fully. So I don't think these people are quacks. I don't think they're people who are intentionally taking advantage of the desperation and desires of others. I think there are people like that. I think these people are true believers. They actually believe that the PRS is really, really strong and they're going to do a good thing for the world by doing this. And I think they're wrong. But there will be, most of life is bell curves. And there will be people and companies who are on the extreme, who will always want to be pushing that envelope. I live in Silicon Valley. I'm surrounded by these people. They'll be there, and it's one of those many areas where caveat emptor, where buyer beware, is really important and really hard, because these are not easy things to figure out, whether what somebody's telling you is solid science or plausible science or really, really speculative science. It can all sound good when it's told in a good spiel to a layman.
Let's take us back to sort of where we are today. And I did see a question in here. I think I lost it about cloning and sort of comparing this to cloning. But it sounds like the way that all three of you have been talking about this, I mean, we're not anywhere near the cloning of humans, right? I mean, we have animals and Dolly the sheep and that kind of thing, but are we anywhere near cloning humans? And would CRISPR be used in any way to do something like that?
You want to talk about mitochondrial disease, the three-parent baby?
Well, we could talk about that one too. I just wrote something earlier, is it this year or last year? This pandemic it's all confused in pandemic time, about the human cloning stuff, which of course, when Dolly was announced, immediately the headlines were about armies of cloned warrior slaves. One sheep, you get people worried about armies of cloned warrior slaves. It turned out that for the next 15 years, nobody could figure out how to clone humans. And cloning turns out to be really odd. It was really hard to do dogs, so that was eventually figured out. It was really easy to do cats. It was easy to do mice. It was hard to do rats. A lot of cattle and a lot of horses. The world's leading polo team uses cloned polo ponies from Argentina. Humans were really hard. And there were a whole bunch of people in the early aughts saying, I'm going to clone humans. There was this guy, Dr. Richard Seed, who looked like the mad scientist from Back to the Future. There was this religious cult called the Raelians, based in Quebec, who said it was a religious obligation for them to clone people. And it all sort of faded out when it turned out nobody could figure out how to do it. Until 2013, when a scientist at the Oregon Health Sciences University named Shukrat Matalapov figured out how to do it. I mean, a major component turns out to be adding caffeine to the culture medium, which I think turns out to be true. So be careful with those double espressos. And he's been able, and other labs have replicated, he can clone human embryos. But we don't hear people, I haven't heard anybody say, I want to clone humans. I'm going to go out and do it. And I actually labeled this little piece I wrote, The Dog That Didn't Bark in the Night, an homage to a Sherlock Holmes story. Why aren't we hearing it? And I actually don't know, other than the fact that cloning never really seemed to make a lot of sense. I mean, if we cloned Rosario, we would get a baby who would look probably a fair amount like Rosario looked as a baby, but would grow up to be really quite different. Similar in some ways, but quite different from Rosario. And with all due respect, Rosario, I don't know why anybody would want to clone an individual. So it turns out it looks like you could do it. Now, it would be crazy to try, right? Because the risks, even with cattle and horses, where it's done fairly routinely, the number of colts and calves born with problems, it's not huge, but it's unacceptable in humans. I mean, to have 5 or 10% of human babies born with fatal issues or serious deformities be a big problem. If you actually took it to the point where you were confident it was safe, we could probably do it. Somebody risky could probably try it now. And it sort of it surprises me that nobody is talking about it. I guess it's just not the flavor of this particular month.
Yeah, interesting. So Michael, maybe take us back to like the here and now, and what do you see in the, kind of near to, I don't know, what would that be, near 5 to 10 years, but, coming up here in the next few years, what sorts of diseases do you think we might see work on this being done? And I guess, should we assume that at least initially it'll be continuing in animal studies before we see anything in people?
Yeah, I've been, the sickle cell disease, I think is the one that Dr. Greeley mentioned earlier. They already have a patient that had treatment and is disease-free. I think she was featured on 60 Minutes a while back. So that's a great example. I think sickle cell disease will really be first and foremost going down the road. I know they're injecting CRISPR constructs for congenital blindness currently. Those trials are ongoing. So we may see a cure for blindness. There was work done at UT Southwestern on Duchenne muscular dystrophy, where they injected CRISPR constructs into the muscle and intravenously, showing a resumption of function in this animal model. So that may not be too far down the road. So I think there will be a growing list of diseases amenable to this CRISPR therapy in the short term. So I think it'll be rapid progression from here on out.
And Michael, would that depend, at least initially, on something that can be fixed with just one gene? Does it get sort of increasingly complicated when you expand that out to 2, 3, multiple genes?
Yeah, I don't think there's a good solution for really complex genetic disorders involving multiple genes at this point. But there are 10,000 single gene mutations out there. So there's a lot of disease that out there that can be cured with CRISPR. 40% of hospitalizations are due to single gene disorders. So there's a lot of need for this type of therapy. So again, if you're going to invest in one tool that's going to make the most difference in the next decade or two, I would put my money on CRISPR.
Wow. And 40%, I mean, we would be talking about just a huge number of individuals and families around the world affected by this. So that's the promising side of all this. Another question, let's maybe go to Rosario for this one. What is the biggest misconception that you hear about this frequently?
The biggest misconception probably is that people think that is ready available, that we can do all the sort of things that Hank and Michael have been explaining, that we can create armies of people, design babies is the most unfortunate term we can use in this setting. I think that's the second biggest misconception is thinking that there's not enough policies or laws regulating this, and the third one is conflating or confusing the ethical analysis in thinking that the right to science versus equity versus diversity, etc. And I think that anybody that opposes germline gene editing, at least at this stage, is very conservative, right? And is very religious, and we are the type that don't believe in abortion and that kind of argument. I think those are for me at least, thinking out of the cuff, 3 of the biggest misconceptions. But maybe others have other comments.
And then on the last one, you're saying that it sort of the abortion debate is so fraught and contentious, and it sort of gets caught up in that maybe unnecessarily. Is that what you're saying? Well,
I always like to think that genetics, but especially this area that the genetic that started with cloning, genetic engineering. So since actually the development of embryonic stem cells have been very embryocentric, right? Overly focused on the human embryo. Maybe right, maybe wrong, but overly focused on the human embryo. And that's have been always confusing the debates about whether the embryo is a human being, whether the embryo is a special entity that deserves a special protection. And from there is the analogies with abortion and the analogies of destroying life in different stages of the research. That is one of the misconceptions, I think, confusing the two things, but also thinking that any ethical argument and scientific argument opposing, at least now, the application of gene editing and germline is because this conservative way of thinking.
Can I add one to Rosario's list?
Yes.
John?
Sure.
So my position in the book is one that guarantees nobody will be happy with me, because I'm drawn to the middle where people at both sides take shots at me. At least it means you don't have to worry which direction the arrows are coming from, because they're coming from every. And I think that germline editing is not necessarily a is not necessarily a bad thing if it's proven to be safe. But I also think it's not a very useful thing in very many contexts. To go back to something Michael said, if a couple that both had cystic fibrosis lived long enough and were healthy enough and they wanted to have a baby together and they wanted the baby not to have cystic fibrosis, germline editing may be the only way they could do that, unless he gets good enough at fetal treatment, which would be another possible alternative. But I think people have this image of the human germline genome in all caps, bold, 18-point red. There is no such thing. I mean, there are 7.5 billion human germline genomes. Each one of us has a different one. Our species doesn't have just one genome. Each one of us has, even identical twins have slightly different genomes because of mutations that happen after their eggs, after their embryos split. And furthermore, they change every generation because of mutations that happen during lifetime. So the gene variations I passed on to my kids were not exactly the ones I got from my parents. And if they ever give us grandkids, they won't have exactly the variations that my wife and I passed on to our kids. And the last point on this, humans have kind of changed what gets inherited by our culture. Agriculture changed human genes. We all have more copies of a gene that digests starch well because our hunter-gatherer ancestors went into agriculture where they had a lot more starch, and people with more starch digesting genes did better. Medicine is regularly keeping alive people who have genetic issues that would otherwise kill them and allowing them to reproduce. So the human germline genome, there are good arguments to be especially worried about it, but it's not the Ark of the Covenant. It's not this sort of sacred single definition of who we are as a species. There may or may not be such a thing, but it's not a genome.
We're getting close to 8:30, and I was thinking maybe we should kind of look to bring this in for a landing. I would like for each of you to maybe give the audience one takeaway, one thing you'd like everybody to understand. And the other thing is, if you could make a prediction about something that you think might happen sometime in the next, I don't know what, few years, that'll be important, groundbreaking, revolutionary, however you want to put it. What's an important development everybody should look for? You want to start with that, Hank?
Well, I think the most important thing that's going to happen with CRISPR, so two answers. I think we should look for eggs and sperm made from stem cells, made from skin cells that are turned into stem cells. I think that's going to happen. My first book, The End of Sex, was about the implications of that. And that will be a big deal, not just for people who are infertile because they don't make sperm or they no longer have eggs. But in the CRISPR world, I think we're too focused on humans. And we are likely to be a little more careful with humans because we are humans. We care about humans. If you've got something that leads to 5% of babies being harmed, that's terrible. If you've got something that leads to 5% of mosquito babies being harmed, people say, why only 5%? So I think CRISPR is already exploding in the non-human world, where we are much more willing to take risks and much more interested in getting corn that deals with heat waves better, or getting mosquitoes that don't transmit malaria from person to person, or getting cats that are hypoallergenic, or making unicorns. And I think we'll use it for all sorts of purposes. And our regulatory system in the US is not ready for that. So the non-human stuff, I think, is much more important than the human stuff.
Michael?
Yeah, as I was saying before, I think what we'll see is an acceleration in diseases that are going to be potentially cured by this technology and within quick succession. So within the next decade, we're going to see a lot more advances. And for me personally, I hope within my career that I'll give parents another option when we make a fetal diagnosis, that we can apply this in utero, potentially prevent lifelong suffering. That's my prediction.
Okay, great. And Rosario, a prediction or a key takeaway you'd like our audience to understand?
Yes, I'm going to leave the audience with a puzzle. I want them to predict the future, and I'm going to give them one key, which is laws, policies, regulation are the reflections of the life, the context, right? What is the social, the cultural, the economic, the historic context? The role of stakeholders, so the role of society acting, the patient activists, etc. If that is the case, then how policies will enable or hinder the application of gene editing in the future will depend on how much this context pushes back or forward. So if I want to predict what will happen, I will say, turn around and look right now at society now. How tolerant this society is to diversity, how tolerant this society is to the disabled community. And when you pass that thermometer, you can make your own prediction of what the future will hold. No law, no oversight is 100% bulletproof. So to predict it, just turn and look at your neighbor. And then you will see how, as a society, we want to evolve or disevolve. I want to leave it there.
All right. I think that's a great point to leave us at. And thank you so much to all three of you. Really appreciate the conversation. And thanks as well to the Center for Bioethics and Humanities at CU Anschutz Medical School, the Denver Museum of Nature and Science, and the Institute for Science and Policy. Thanks so much. And I hope everybody has a great evening.
Thank you.
Thank you.
Bye all.
Thank you. That was fun. Bye.
Thank you.